Hello everyone, my name is Elric Esposito and I will be joined by Julien Fernandes. Today we are going to talk to you about the Dual Inverted Single Plane Imaging Microscope or DiSPIM and present you the system that is installed at the Photonic Bioimaging facility of the Institut Pasteur in Paris. Here is a summary of what we'll be covering in this talk. We'll do a general system presentation, then we'll talk about sample mounting, system operation, post-acquisition treatment, and finally some tips on how to use the system. General aspects of light sheet modalities have been covered elsewhere and won't be treated here, so we'll be focusing on this particular microscope itself. So let's begin with the system presentation, starting with its geometry. This system has been introduced by the group of Harry Schroff in 2013 in order to address high-speed, high-resolution 4D imaging on flat-mounted samples. This setup can be mounted on a classical inverted microscope system. In order to work on flat-mounted samples, the microscope itself it's tilted at 45 degrees in a V-shaped format. It is composed of two identical arms, including laser delivery and bidirectional scanning to generate the light sheet for illumination and a camera for detection of the opposite side. This dual illumination allows acquiring two views of the same plane by quickly alternating a detection arm into an illumination arm. This dual view provides isotropic resolution upon post-acquisition treatment. Several scanning modalities can be performed with this light sheet microscope. First, we'll be covering what we'll call slice scan. The 2D scanner scans a focused laser light beam in one direction in order to generate a single plane illumination that will be detected at 90 degrees by an SMOS camera. As both objectives are mounted on piezo stages, it can follow the light sheet beam as it is scanned through the sample. In an attempt to clarify the geometry of the system, here is an alternative view illustrating how the camera thinks through the objective. Once the whole volume has been acquired on one side of the sample, each arm can switch from illumination to detection and vice versa, acquiring the same volume on a different angle. We'll call path A the view where the beam illumination comes from the left-hand side and detection is done on the right-hand side, and path B the one where the illumination comes from the right-hand side and detection is done on the left-hand side. The volume that can be acquired in this fashion is limited by the light penetration depth and the range of the piezo stage, as well as the field of view of the objective. In our case, that corresponds to 150 by 330 by 330 microns for a 40x objective using the full camera sensor size. An alternate way is possible by scanning the sample through a fixed light sheet. This is what we'll call stage scan. This method allows you to increase the range of acquisition, enabling millimeter-sized imaging such as a zebrafish or an embryo. In this animation, only one side is illustrated for clarity purposes, but the sample goes through both light sheets. As before, each plane is illuminated twice before the stepper motor stage moves to its next location horizontally. To our knowledge, there are several ways to get your hands on such a system. You can build it yourself following the instruction from the Schroff's group paper and the very complete DiSPIM wiki. Most of the mechanical parts can be provided by ESI and some can be 3D printed. This obviously gives you the optimum flexibility to match your particular experiment. You could go for a fully closed system as provided by the Luxendo line from Booker. Their QV system has a similar geometry to the DiSPIM, and it is thought to be a turnkey solution that removes the optical adjustments from the user side. The system we have is something in between. The microscope we'll use here is the Mariana light sheet from 3i. Although all you see here has been done with the water dipping 40x objective shown here, you can field alternate objectives such as a 10x 0.3 NA for a higher field of view. The 40x 0.8 NA is our go-to objective as it allows us to be the closest to the Nyquist criterion for the 6.5 micron pixel camera. Speaking of which, our DiSPIM is fitted with two Orcla Flash 4 SMOS camera from Hamamatsu, 4 laser lines, and is fully incubated with CO2 and temperature control for experiment on live samples. Fluorescence detections occur through quad band filters in front of each camera. The system is driven by the software slidebook that we'll also use for data visualization as well as post-acquisition treatment. Moving on to sample mounting, I'll let Julien describe the chamber and the sample preparation.
First, we have the chamber. It's made in three parts. The first part is the bottom part, which is the holder for the cover slip. The second part is the top part, which contains the silicone joint, allowing the chamber to be waterproof. Third, you have the screws. For this chamber, we use the cover slip 24 by 50 with a thickness of 0.17. To mount the chamber, we put the cover slip in the notch made for the cover slip. Be careful to insert well the cover slip into the notch or you may break it. Put the top part over the cover slip and screw the 4 screws one after the other one, gradually until it becomes waterproof. You can test the tightness by putting some water into the tank. Once it's completely assembled, this chamber fits into the O-collab support and then it's covered with the cover adapted for the geometry of the DiSPIM. As Elric mentioned in the introduction, the geometry of the DiSPIM enables to mount your sample on a conventional cover slip. This simplifies the usual cumbersome mounting of the sample that usually comes with the light sheet microscopy. As far as the sample preparation is concerned, the video showed here describes the mounting of a root embedded in an echographic gel and then immersed in water. This stainless steel chamber can be autoclaved and used for in vitro, in vivo, or ex vivo experiments. A general mounting principle that can be applied to pretty much all types of samples to be imaged in this system consists in an adhesive layer holding the object to be viewed in place and covering up with media where the objective will be dipped in. Given the mechanical dimension of the chamber, particular care needs to be taken to position it as close to the center of the slide as possible. This will prevent the reduction of the observation field due to the objective bumping into the side of the chamber. In order to increase the freedom of movement of the chamber, other large inserts may be inserted, such as an 85 mm diameter Petri dish.
Now that the system has been described and its mounting has been covered, we can move on to system operation, starting with pre-alignment. The system has been powered up and the chamber is currently filled with a fluorescent solution. The scanner is off so that we have a single beam on each side going through the sample. Both objective piezos are set to mid-range position and we are adjusting them so that both beams are in focus. We hit beam for a single beam configuration on both channels and we can see both arms alternating from illumination to detection. Each beam corresponds to a different path and is assigned a color, red for path A and green for path B. By the way, apologies to colorblind folks watching this video. There is an option to switch to cyan and yellow, but I forgot to switch that on when I recorded this video. We can separate the two beams by sliding one objective aside. Aperture at the back of the objective have been closed so that we end up with a pencil beam. In other words, the depth of focus has been set to cover the field of view of our objective, which is 330 microns in our case with a 40x objective, using the full camera chip. Physical adjustments are made by screwing the objective in and out so that we have a nice focus beam at the end of the process. Once this is done, we put the two beams back together using the slider again. We now need to check that the center of a stimulation is the center of our camera. If this is not center, we can adjust the tilting screws on the corresponding beam to align it properly. Now that our beams are aligned, we can activate the galvos in order to generate our light sheet by pressing a button. We can stop the switching from one path to the other and focus on a single path like so. Adjustments on the scanner allows to control the sheet's offset between themselves and their width. We can crop the camera sensor to refrain recording more data than we should. A single point can easily reach several gigabytes as light sheet dataset can be quite computer heavy. Therefore, we want to make sure that we only record what is necessary in order to save processing time down the line. At the end of this process, we want a nice homogeneous use single plane illumination that will cover our full field of view. It is not necessary to systematically go through this before each experiment, but it is easy enough to check in order to avoid surprises with the actual sample. Our system is set now, and we can introduce our sample. For this demonstration, we'll be using a live zebrafish embryo genetically encoded with GFP. We can use the inverted microscope to locate and position the sample. We can also use standard AP wide-field microscope to check its fluorescence. Again, we are now looking at the sample from underneath. Once this is done, we can drop our DiSPIM objectives and search for it in light sheet mode. We are now live with both our light sheets alternating from path A to B with excitation light at 488 nanometer. We carefully lower both objectives using the stepper motor in order to avoid smashing the sample and cover slip with the bulky objectives. The top of our zebrafish comes into focus. Both images from both paths are overlaid on the live window. Again, here, red and green colors assigned corresponds to the image produced by its respective path. All fluorescence detected comes from the GFP. Due to the system geometry and its 45-degree rotation, it is important to realize that when moving the sample stage horizontally, you are effectively scanning the sample in Z as well, as illustrated in this animation where several heads are represented. Reaching the region of interest implies adjustment on the XY stage and vertical adjustments on the objectives arm. We now need to perform a piezo calibration on the sample that will define our working range. To do so, we will work on each path independently, starting with path A. Starting from a mid-range position, we induce a 70 micron shift to the objective piezo upward and then adjust the light sheet position so that we have a focused image for this objective position. We then move in the other direction from the center with a minus 70 micron shift and again finding the right light sheet position for the second extreme. This will allow the system to have an accurate objective piezo position as the light sheet slices through the sample. We can check that all is in order by setting the objective to a central position and clicking Adjust. And the system will compute the correct light sheet corresponding position. We can see that the image is in focus, hence the calibration correct for this path, and we can move on to path B. Our slicing range has been calibrated for 140 micron movement on the piezo. The procedure is identical for path B. Again, we are shifting the objective to an extreme position and match the light sheet position. So that's plus 70 that would arrive around here. So we set that, then shift to minus 70 and again find the right position, which should be around here. Once this is done, we center both objectives and click Adjust to check that all is good. The calibration is now complete and we are now ready to proceed for acquiring our sample. We'll first demonstrate acquisition using slide scan. Once we have our area of interest, we can select our sensor cropping factor. As we are using the 40x 0.8 objective with a 6.5 pixel size camera, we are close to the Nyquist criterion. Cropping the sensor size from 248 to 1040 pixels square, we provide a field of view close to 160 micron, which is a good compromise between speed, field of view, and resolution. Then we select the number of planes and our step size. Here we choose 300 planes with a step size of 0.5 microns to exploit the full range of the piezos. Although only GFP is present in our sample, we could set extra stimulation wavelength in the advanced menu for multicolor imaging. We are now set and can execute the acquisition of our volume. The video is in real time for an acquisition volume of 160 by 160 by 150 micron with a voxel size of 160 by 160 by 500 nanometers. It's obviously preferable if the fish does not decide to wiggle at the beginning of the acquisition during a screencast recording, but I guess that's the beauty of live sample imaging. Acquisition is now done, and we are going through our stacks. I am again changing the lookup table to discriminate between the images originating from path A and B. We can also observe the volume acquired in 3D as you would in a typical visualization software. Each channel is assigned to one of the paths. We can see that the image is 1.2 gigabyte for one time point single wavelength excitation, despite our efforts to reduce it as much as possible. We now move on to stage scan modality, and we still have our zebrafish sample. Remember that when we are scanning the sample across the light sheet, we are effectively scanning through various depths. We'll cover that in more details in the next section. For now, all we have to do is select stage scan in the software menu, select the step size, and define two extreme positions. This is what I'm doing now. Moving all the way to the left-hand side, set this position, and then go all the way across to the furthest position on the right. We can then check the central position for signal adjustments if necessary. As the sample is going through fixed light sheets, we can crop the sensor size even further in order to avoid redundant data. As before, we are now recording in real time a volume of 680 by 163 by 30 micron. This should take less than a minute and benefits from the good fluorescent signal coming from the sample, allowing us to work at 25 millisecond exposure time. Acquisition in slice scan is done. This demonstrates the high-speed acquisition capability of this system for this relatively large volume. We can go through the acquire stack as we did before, and we'll discuss mathematical treatment that needs to be done for 3D view in the next section. So let's now look at post-acquisition treatment. Let's consider the data we just acquired in stage CAD mode. First, we see that a 90-degree rotation must be applied to one of the channels for both their stack from path A and B to be overlapped. This can easily be understood given the right-angle system geometry. Trying to render the acquired image in a classical software becomes challenging as images are a succession of parallel planes at 45 degrees angle. If we stack those images together, we would end up with an incorrect geometrical representation when aligning the images side to side, as you would with a deck of cards. The process of an accurate geometrical display is called DiSPIM. As it knows the microscope geometry, the software correctly computes and renders the volume acquired in stage scan mode and produces a conventional Z stack that could be opened and viewed in a classical 3D visualization software. Extra voxels are generated to fill the gap. The final 3D image still appears rotated compared to a conventional Z stack that one could get from a laser scanning confocal. The cover slip function allows to correct for that and visualize the data set as if it was coming from a confocal. All images generated on our system can be imported in other softwares for customized channel transforms via the bioformat standard. The purpose of this dual-arm geometry is to enable isometric resolution, thanks to its dual view of the same illumination plane. We still have one more step to follow before that, which is image registration. To illustrate this, we'll move away from our zebrafish sample and we'll use this organoid acquired in slide scan mode. The process we'll describe here will be the same for stage scan acquisition. Again, we have red assigned to path A and green assigned to path B, and common features can be identified. We can clearly see that they do not overlap. Slidebook allows us to do a pretty straightforward registration procedure as far as the end user is concerned, using a cross-correlation method. All we have to do is compute the background and let the system compute initial shift and apply it to our stacks. Pre-alignment that we covered earlier on and good signal-to-noise ratio helps the system along to come quickly to an acceptable solution, as shown here. The process is relatively fast, but is sped up in this video. Both stacks appear nicely overlapped now compared to earlier on. We can rotate the sample to check for inconsistencies before validation on the on the computed matrix shift coordinates. We can also compare the stacks with and without registration. The registration part is now completed, and we can move on to the final part of our DiSPIM image processing, which is image fusion and deconvolution. We'll use our previous organoid that has been correctly registered, and we now need to define a bounding box that will contain the data to be worked on by the algorithm and exclude what is not necessary. A nice feature from Slidebook is the ability to dim the data that is outside the box so that you can have a more accurate visual feedback of what you're doing. We need to make sure that our box includes all that we need on all axes. Once I'm happy with X and Y, I move on to a different view on the Z-axis in order to fit all in. The box is now set, and I can apply that and assign this box to this dataset. Once we have done that, we need PSF data for the deconvolution. We have previously acquired nanometric beads with diameter inferior to the objective diffraction limit, which is 200 nanometers in our case. We need to isolate one bead acquired on both paths inside what would be our PSF bounding box. This will be used in the deconvolution process. We can also point to a file containing a single bead. We are now showing the process of selecting one bead for demo purposes, but we will be using a previously saved file in the actual process in this video. So I am now closing my PSF box to a single bead. And when this is done, I can save that for later. Now going on with the actual process, we select multi-view reconstruction deconvolution in Slidebook, and we are presented with this menu. We check that the files has bounding box information and registration coordinates. We only have one time point here, but we could obviously select how many we'd like to process. We then compute the background, select our PSF file that we recorded earlier on, choose the number of iterations and our algorithm type. We can split the data to fully exploit the graphic processor unit and speed up the process. All seems good, so let's apply that. Depending on the dataset and the computer specs, this process can be quite long, for example, a few hours for 10 to 12 hours long experiment with our machine. On this dataset, this is rather fast, and the decomposed image is generated on a separate file. So here is our fused image. As you can see, only one channel is present now, and is a fusion of path A and path B together. This organoid is a 160 micron side cube at subcellular resolution that has been acquired under 45 seconds. We can see that it appears evenly illuminated and has isotropic resolution. Before wrapping up, we'd like to give you a few tips that we hope you'll find helpful if you'll be using this system. We would advise to center the sample in the chamber the best you can. This will avoid to lose imaging range due to the objective bumping into the side of the chamber. Better reconstruction will happen if the signal levels are even on both paths. It is possible on slidebook to balance the laser power and have a live histogram in order to achieve that. Be aware of evaporation at 37 degrees. Even with the chamber on top, we seldomly experience evaporation on overnight experiments. This results in the objectives not being dipped into the water, therefore the sample becoming out of focus. Hence, do not hesitate to fill the chamber with as much liquid as possible for overnight experiments at 37 degrees. Using gel in our hand was not very successful. We have a good computer, as we've seen that dataset can become rather bulky. As an example, here are the main specs of the computer we've used for this presentation. In summary, DiSPIM is an interesting system if you plan to work on flat sample geometry. As all light sheet microscope, you can expect fast acquisition and low phototoxicity. Post-processing allows subcellular resolution on relatively large samples. However, for some applications, having quad band filters may become an issue if you have fluorescent crosstalk. You need to be aware of the depth that you can reach with the objectives due to mechanical constraints. This system can be intimidating to an experienced user due to the pre-alignment procedure. Currently, our system does not allow multi-point scanning, which would be highly beneficial for some experiments, but it is our understanding that this is being under development by the manufacturing companies and ought to arrive soon. This concludes this video. We'd like to thank the people of the imaging platform at Pasteur, our colleagues that provided the samples, and the 3i engineers that have trained us to use it. Many thanks also to the funding bodies listed here, with a special one to the Région Île-de-France that funded a big part of the system through the GMLC grant. Finally, we'd like to thank you for your attention.